PAL.
Computational identification of natural compounds as PARP1 inhibitors for triple-negative breast cancer treatment. Hey everyone! Welcome back to our channel. Today, we're diving into a fascinating topic that's making waves in the medical research community: the use of indole-based natural compounds as potential inhibitors of PARP1 in the fight against triple-negative breast cancer, or TNBC for short, published in the Journal of Biomolecular Structure and Dynamics. Now, if you're not familiar with TNBC, let's break it down. Triple-negative breast cancer is one of the most aggressive forms of breast cancer. It's called triple-negative because it lacks three key receptors: estrogen, progesterone, and the human epidermal growth factor receptor 2, or HER2. This absence makes TNBC particularly challenging to treat, as it doesn't respond to many common therapies. So, what role does PARP1 play in this? Well, PARP1 is a protein that helps repair DNA damage in cells. Unfortunately, cancer cells can exploit this repair mechanism to survive and proliferate. This is where PARP inhibitors come into play. By blocking PARP1, these inhibitors can prevent cancer cells from repairing their DNA, ultimately leading to cell death. In a recent computational study, researchers screened a whopping 200,000 natural products from the Universal Natural Product Database. They were on the hunt for potential PARP inhibitors. Using a technique called molecular docking, they identified 6 promising compounds that showed strong binding affinity to PARP1. Now, let's talk about what binding affinity means. In simple terms, it's a measure of how well a compound can attach itself to a target protein, in this case, PARP1. The stronger the binding, the more effective the compound is likely to be as a drug. The researchers didn't stop there. They also performed ADMET analysis, which stands for absorption, distribution, metabolism, excretion, and toxicity. This analysis helps to evaluate whether these compounds have the right properties to be developed into safe and effective drugs. They looked at factors like bioavailability, how well a drug is absorbed into the bloodstream, and potential toxicity, which is crucial in ensuring that any new treatment won't cause harm to patients. Next up, they conducted molecular dynamics simulations over a period of 200 nanoseconds. This is a fancy way of saying they used computer models to simulate how the protein-ligand complexes behave over time. They wanted to see if these compounds remained stable and effective in binding to PARP1 throughout the simulation. The results were promising. 2 compounds, referred to as HIT-3 and HIT-5, exhibited stronger binding energies than talazoparib, an FDA-approved PARP inhibitor. Binding energy is essentially a measure of how energetically favorable the interaction is between the compound and the target protein. The more negative the binding energy, the stronger the interaction. During the study, researchers observed strong interactions between these compounds and specific hotspot residues in PARP1. These are key areas on the protein that facilitate binding. The residues involved included ASP770, ALA880, and TIR896, among others. This highlights the importance of these interactions in the overall effectiveness of the inhibitors. Let's look at the binding scores. These scores indicate how strongly each compound interacts with PARP1. HIT-1 had the highest score at approximately -19.94 kcal/mol, followed closely by HIT-2 and HIT-3 at -18.85 and -18.64 kcal/mol, respectively. The negative values here are significant. They suggest that the binding is energetically favorable, which is a good sign for potential drug development. Now, what's really interesting is how these compounds interact with specific residues in the PARP1 protein. For instance, HIT-1 forms strong hydrogen bonds with 2 important residues, TIR896 and ARG878. These bonds were measured at distances of just 1.7 and 2.9 A, which indicates a very close interaction. Hydrogen bonds are like tiny magnets that hold molecules together, and the stronger these interactions, the more likely the compound will effectively inhibit PARP1. This is crucial because the stronger the binding, the better the chances of blocking the protein's activity, which could lead to reduced cancer cell proliferation. Moving on to the computational methods used in this study, the researchers also employed a technique called PBSA, which stands for Molecular Mechanics Poisson-Boltzmann Surface Area. This method helps calculate the binding free energy of the protein-ligand complexes. In simple terms, it allows scientists to estimate how stable the interaction between the compound and the protein is. They calculated the binding free energy using 100 snapshots taken from a 200 nanoseconds molecular dynamics simulation. This simulation helps visualize how the compounds behave over time in a dynamic environment, mimicking real-life conditions inside the body. The results showed that HIT-3 and HIT-5 had even stronger binding energies than the FDA-approved PARP inhibitor, talazoparib, with scores of -25.64 and -23.14 kcal/mol, respectively. Talazoparib, for reference, had a binding energy of -10.74 kcal/mol. This comparison is vital because it suggests that these natural compounds could be more effective than existing treatments. But wait, there's more. The researchers didn't stop there. They also performed a per-residue energy contribution analysis, which helps identify how much each amino acid in the protein contributes to the overall binding energy. This analysis revealed that several hotspot residues, such as ASP770, ALA880, and TIR896, played significant roles in the binding interactions. Understanding these interactions at such a detailed level can help in designing even better inhibitors in the future. So, what does all this mean for the future of TNBC treatment? Well, these findings provide critical insights into potential new therapies that could be developed from these natural compounds. By targeting PARP1 more effectively, we could see advancements in how we treat this aggressive form of breast cancer. To sum it up, the research highlights the potential of indole-based natural compounds as promising candidates for PARP1 inhibition in triple-negative breast cancer. With ongoing studies and advancements in computational methods, we hope these findings could lead to new, effective treatments for patients battling this challenging disease. If you found this information helpful, don't forget to like, subscribe, and hit that notification bell for more updates on the latest in medical research and breakthroughs. Thanks for watching, and we'll see you in the next video.
PharmaPal.